Come and revise GCSE biology with me. How would you carry out an experiment to determine the impact of light intensity on the distribution of some clover plants? Hi, my name's Dr. De Bruin. I'm a GCSE and A-level science teacher, and I also make revision videos on YouTube. This is one of the 2 required practicals in Combined Science Biology Paper 2, and we know that those required practicals make up 15% of all the marks available, so they should really be your number 1 priority for revision. In this sampling required practical, you're going to have to use a quadrat. This is a square or rectangular frame with or without internal divisions that you're going to put down in different places and see is the clover there or not. Depending on the exact wording of the question, you may be doing random sampling or transect sampling. You use transect sampling if you're moving from an area where it's dark to an area where it's light. Maybe you start out underneath a big tree and you're going out into the... So you use a tape measure to make one long straight line, and you put your quadrat down maybe every 5 metres or every 10 metres and assess the abundance of the clover at each quadrat. We use random sampling if you've got a couple of different locations, one with very high light and one with lower light, and you're using the random sampling to take multiple samples in each location. Now, random sampling does not mean shut your eyes and throw it over your shoulder. For one thing, that's not super safe, and for another, it's not random. For random sampling, you're going to take 2 tape measures and basically make yourself a grid. Number the possible locations, say 1 to 100, and then use a random number generator to tell you which of those quadrats you're going to sample. Now, there are lots of different versions of this practical. I said we were going to investigate light intensity, so we would need to use a light meter. But you could also investigate pH using a pH probe, or water availability using a moisture probe. And whichever abiotic factor you're investigating, every time you put your quadrat down to count your species, then you're also going to measure that abiotic factor. You might be asked to work out a mean abundance for the species in the whole field. So in that case, firstly, we'd work out what the area of one quadrat. Usually they're a metre by a metre, so just a metre squared, but it could also be half a metre by half a metre, so 0.25 metres. Either way, you average out all of your quadrats for a particular area and work out what the mean abundance for 1 metre squared is. And then you look at the total area for your total sample area and multiply it up. So say if we had 12 plants in a 0.25 metre squared, then our mean coverage for 1 metre squared would be 48. And if we had 200 metres squared of field, then 48 times 200, we'd be estimating that we have 9,600 clover plants, I think.